Thank you very much. Good morning to everybody in Asia and good evening to all of you in the Americas. It's great to be here. In the next few slides, I'm just going to give you a sense of how far we've come from the basic microRNA research that started a little over 20 years ago to now being in the commercialization stage of this exciting area of science. So, as was mentioned, I have a few disclosures. I'm a founder of certain companies, and I'm also on the scientific advisory board for Merixis. And it's great for me to be here. If you could go to the next slide, please. Do I work the slides? Can somebody go to the next slide? There we go. Can you go back one? And back again? Okay, forward one. Thank you very much. So, as John was just saying, RNA is, of course, a central player in biology, but it's only relatively recently, over the last 20 years or so, that we've fully appreciated just how extensive RNA reach is in our cells. We all know about the 20,000 or so protein-coding messenger RNAs that our cells have the potential to encode. But what was not apparent even 20 years ago was that we have almost 10 times as many non-coding RNAs that are made from our cells. And these non-coding RNAs play regulatory functions. So, of course, they don't code for proteins. And one of the most famous examples of these non-coding RNAs are the microRNAs. MicroRNAs, as John mentioned, have the ability to base pair with other RNAs like messenger RNAs and regulate their expression. So microRNAs were discovered only about 20 years ago. And as I'll show you in the next few slides, we've come a very long way from discovering a completely new class of regulatory molecules in human cells to now finding their utility in clinical trials and as potential therapeutics as well. So next slide, please. So while microRNAs are small RNAs, John mentioned they're only in the 20 to 22 nucleotides size range. They're extremely powerful. So they play roles in almost every part of our development, our homeostasis, our aging, and of course in many disease states. MicroRNAs are not only found in animals, but also found in plants. And so there are now agricultural applications that are also emerging in plant biology. So one of the things that we'll talk about mostly today is on the bottom right there, the early detection saves lives part. So microRNAs, as John mentioned, are extremely stable within bodily fluids, including serum, blood, tears, urine, etc., and are increasingly being used in diagnostic applications. So next slide, please. John also mentioned this slide, but I'm going to just go into a little bit more detail. John mentioned that microRNAs play multiple roles in cancer development. In almost all of the stages, all of the hallmarks of cancer, we can find particular microRNAs. So the one microRNA that we discovered many years ago is LET7. You can see it there on the sort of at about 4 o'clock on the wheel there. But another microRNA that we worked on quite extensively is miR34 at about 5 o'clock. And we and others have shown, for example, just as an example, that miR34 regulates the expression of PD-L1, which is, of course, one of the immune checkpoint genes that is targeted in immune checkpoint therapy. So another thing that John mentioned was that these microRNAs, while they have functions within cells, they also get packaged into exosomes. So on the right, we're showing these little microvesicles and exosomes that can package a variety of different RNAs, including microRNAs. And these microRNAs can travel through the bloodstream to actually affect the activity of other cells. So next slide, please. So this is an illustration of how a cell can sort of make microRNAs and then package them into these vesicles, and then the vesicles can enter the bloodstream, shown there in the middle, and exit the bloodstream at some distant point in the body and into cells and regulate gene expression within those cells. Next slide, please. So because microRNAs are found in many of our body fluids, they are emerging as attractive biomarkers of the sort of type like circulating tumor DNA or circulating tumor cells. However, one thing that we and others have noticed is that while circulating tumor cells and circulating tumor DNA are found at very low levels within the blood, microRNAs can be found at very high levels and are easy to detect within the blood. As John mentioned, they're very stable. They're packaged in vesicles, or they're incorporated into the Argonaut risk complex, and they can survive harsh conditions in the blood. They can also survive harsh conditions of freezing and thawing and fixation, and so microRNAs can be detected in retrospective studies of samples that have been frozen or fixated decades ago. So next slide, please. So while we discovered the first human microRNA in Gary Rifkin's lab back in the year 2000, it was only in the year 2008, in a big surprise, that groups were able to show that microRNAs could be detected in blood. And at about the same time as that happened, a number of companies were starting to develop microRNA-based diagnostics for prognosis or early detection. The first one was launched by a company called Rosetta, and it allowed physicians to determine the site of origin of a tumor that had caused a metastasis where it wasn't clear where the metastasis had actually arisen from. Over the course of the next 10 years or so, a number of governmental and other organizations launched programs in trying to sort of translate these discoveries into new biomarkers, including in Singapore, in the United States, and in Japan. In 2014, the first insurance reimbursable test based on a microRNA came out from Interpace. This was a microRNA-based assay that could detect thyroid cancer within patients. And since then, we've seen a number of different microRNA panels being approved, mostly in Asia. At this point, there are no microRNA-based panels that are of sort of multiple microRNAs in signatures that have been approved yet in the United States, although it probably won't be long before that happens. As I'll show in the next slide, there are a number of different clinical trials that are happening in this space, and so it surely won't be long. So one reason why I'm excited about this particular launch tonight, this particular standards, and why I got involved, is because I've been working in the space a long time, and I've realized just how variable everybody's assays are, and just how tightly controlled one needs to perform these sorts of experiments. There's variability that's introduced by how the sample is collected, how the sample is stored, how long it's stored, what type of tissue one generates the RNAs from, and then which assay one uses to detect the RNA, and then also how one standardizes to sort of normalize expression. And so with Mirexis, I've been very excited to sort of work with them to try and develop a standard that would be applicable throughout the world. And I'm very happy to see that the standard has been approved in Singapore, and that it's now being discussed at the international level as well. And so another reason why I'm so excited is if you look in the next slide. Next slide, please. If you just do a search through clinicaltrials.gov, which is a database of all the clinical trials that are happening around the world, and you search for clinical trials involving microRNAs, well, you can see there in the top middle that there are over 900 clinical trials at the moment involving microRNAs mostly as diagnostic biomarkers, but there are others that are involving microRNAs as therapeutic targets as well. And so we're probably on the cusp of seeing a number of these biomarker panels being approved for a variety of different cancer and other disease indications, especially for early detection, but also, as was mentioned, for prognosis and for predictive biomarkers. And it's going to be increasingly important that we trust the veracity of the assays and we can trust the data, especially certain companies that are moving these assays into clinical trials. They would almost certainly want to make certain that the assay is robust and that they're getting the best possible data from each sample. And so with that, I'm going to close and just say congratulations to the team for getting this together, and I look forward to your standard being applied more broadly. Thanks everybody.